Thank you so much for this opportunity to share my recent work. I'll share my screen for the PowerPoint presentation, which is about the biosocial entanglements of foods, epigenomes, microbiomes, and environments in modern human becomings and health. I just have to start here. Yeah. So anyone can see clearly?
Yes, I mean, I can see it.
Yeah, okay.
Yes.
Good. There is one thing in the, yeah. Okay, so we are actually living in a new era of the biosocial genome. It is said where biology is being acknowledged as being shaped through an ongoing situated process. The timeless, universal, and non-responsive autonomous genome sequence is now being supplanted by the environmentally responsive epigenome and microbiome. This scientific evolution heralds a less deterministic and more relational biology, where molecules, microbes, and human bodies are co-constituted through heterogeneous environmental influences, be it socio-cultural, natural, economic. The responsiveness of both the epigenetic molecular regulation of gene expression and the different microbial communities, as well as their genetic material, which is called the microbiome, vis-a-vis social-cultural factors, is what excites people like me with a background in social science. Because we see herein the possibility to mend the historic nature-nurture divide where biological determinism used to reign free. Yet, despite these promises and hopes, such rapprochement is not evident, as it requires thorough interdisciplinary cooperation at a practical level and moving beyond the habitual ontological and epistemological positions of the disciplines involved. So I suggest that food is actually a key facilitator for such radical interdisciplinary cooperation, provided that food is carefully conceptualized, as it enables tracing the biosocial or material semiotic network of food and food systems and their multiple heterogeneous entanglements with, on the one hand, socio-cultural organization of life, and on the other, the microbial and molecular regulation of life on the other hand. As food is key to health, we'll also discuss implications of this approach for a broadened conceptualization of health that is less anthropocentric, but precisely is attentive to the human and other than human beings entangled in what I call the more than human being, or rather human becoming. I will gradually build a programmatic agenda by discussing my attempts as being a predominantly social anthropologist toward a radical interdisciplinary cooperation with researchers in epigenetics and gut microbiota, and the lessons I've learned on the way in this endeavor. I actually started as specializing myself in the anthropology of food and eating and food systems during my PhD here at CLEA. I conducted research in Sri Lanka following all the lifelines of food from seed to stool, and how food during its life course entangles with and takes part in shaping different realms of life: domestic life, village relations, cosmological orders, politics, and so forth. And given this very large multiplicity of articulations, both meant to entail expression and connection of the same food in different contexts, For instance, the coconut used as a ritual agent of healing versus the coconut as an economic commodity. Following all these things, I conceptualized food as an assemblage of heterogeneous components with which it is entangled and is engaged in a relation of mutual becoming. In other words, being entangled. This parallels the notion of food being a material semiotic network where parts acquire form and meaning in relation to each other in a specific context, just as a word acquires meaning in a sentence. This multiplicity in potentiality turns food into a powerful agent with which to work in learning and shaping society and health. And this across multiple scales, ranging from the gut to the globe. During my postdoc, I delved deeper into the intimate entanglement of food and health as conceptualized in both local health traditions in Sri Lanka and biomedicine. And besides delving into the elements and the humoral theories of the eco-health system that Ayurveda and other Asian health systems are, it was also during this research that I started reading about epigenetics and especially environmental and nutritional epigenetics. But before continuing, let me just briefly explain epigenetics for our purposes. It is actually the study of the molecular regulation of gene expression, either enabling or hindering a gene to mark a phenotype, which is the observable characteristics, without changes in the DNA sequences, because that would be a mutation. There are three main mechanisms of gene regulation studied by epigenetics. And the first is the most well-studied one, DNA methylation, where methyl groups attach to both coding and non-coding regions of the DNA, affecting the transcription to messengers and messenger RNAs that get translated into proteins that shape the phenotype. Other mechanisms involve histone modification or acetylation, where acetyl groups change the histone's shape and that affect the chromatin structure and accessibility of DNA for transcription. Third, we have the non-coding RNAs that are not transcribed to proteins and that nevertheless play a regulatory role. So these three mechanisms, they also interact with each other in opening and closing DNA for transcription, that is, for its actualization into a phenotype. And these changes get transferred either mitotically to other cells or meiotically in cell division, which leads to different forms of heritability, some more debatable than others. The subfield of environmental epigenetics is the field that studies environmental exposure or signals and that seeks to trace, and I quote Landecker and Panofsky here, the impact of environmental factors on molecular processes, then the ensuing shifts in gene transcription and translation, and then the subsequent physiological or behavioral changes. End quote. In other words, it chases complex chains of causation, molecule by molecule, from the outside and the inside and back. And so as food has become both environment and signal in current research, an important subset of environmental epigenetics is precisely nutritional epigenetics. And as a food anthropologist in origin, that of course raises my interest. So nutritional epigenetics is often also related to the big field in medicine, the developmental origins of health and disease, where then food is approached as a signal in utero that shapes in anticipation to the outside world in which you will be born, the body of the infant and its metabolic system to process food in the future. So the metabolic system of the child is also an anticipatory system in utero. So we see thus that reactive molecular regulation of genes hold a salient promise for reconnecting the biological and the socio-cultural aspects, thereby bridging the discipline-based nature-nurture divide. But more importantly, it also allows us to go for a more processual and situated biology, where the human body is in a constant state of co-evolution. In other words, human being is actually always a human becoming in interactions with the environment in the broad, diverse biosocial sense. Yet, the tendency in epigenetics is all too often to molecularize the environment and to narrow it down to proximal explanations, and that constitutes a key challenge in developing a more integrated research or an approach. Anyhow, I started looking for the rare, relevant research that moves precisely into the direction of intercultural epigenetics, a bit a la Eva Jablonka, and Hannah Landecker, who is the director of the UCLA Center for Society and Genetics, alerted me to a very interesting study on colon cancer in the Middle East, carried out by Chan and his team. And they found that the epigenetic patterns of colon cancer were different across Egypt, Jordan, and Turkey, indicating that a dissimilar molecular pathogenesis exists, and thus that colon cancer develops in different ways. For instance, the age pattern differs, and also they detected different CpG sites on the DNA that were methylated. And in that, and also the location where the cancer emerged. And in the molecular characteristics of the Turkish colon cancer, it was more like in the same location as the Western colon cancer, in the right side location, and whereas the colon cancer emerged in more rectal areas in Egypt and Jordan. So they suggest that this differential molecular pathogenesis points at different so-called environmental exposures, and particularly food habits, because they see that the Turkish lifestyle is a bit closer to the Western lifestyle, and therefore the molecular pathogenesis is more similar in Turkey than in the two other countries. So I think, given that environment is so key, the concept of salutogenesis of Aaron Antonovsky may be useful to think this further. But in my personal wayfaring in these new fields, I contacted Wim Van Daele, a nutritional epigeneticist in Belgium, and we developed a research proposal together with Hannah Landecker, where we would try to craft a project to measure the epigenetic impacts of food habits. And in this way, our attempt was to trace biosocial causative pathways between food culture and the broader social context on the one hand, and the molecular regulation of gene expression on the other, exploring these through tracing the assemblage network of food and its entanglements. It was all nice in theory, but the practical elaboration of this project proved to be challenging. On the one hand, for the epigeneticists, it is important to have a very large sample of people to be able to find statistically relevant variations, to be able to make a clear statement about the population-specific variation in the epigenome. Yet, it is very impossible to have a sample of, let's say, a few thousand of people and then combine it with the anthropological requirement to have a very detailed collection of biographies of people, events, and habits, recording every day what they eat. So doing that for thousands of people, then it's really impossible. Yet, as we know, the epigenome is so reactive that all these different small events do have an impact. So the amounts of data to fulfill both disciplines' demands in terms of an adequate sample size for the epigenetic science, as well as the in-depth details of lived reality, seem to be impossible to be combined for a single fundable proposal. So then we came up with working through principal component analysis, working with which is a statistical technique to reduce the noise of having enormous multidimensional data sets with a minimal loss of data and to facilitate interpretation. So that would have enabled us to tease out the epigenetic factors that lead to the highest variance and that are uncorrelated with each other. So this principal component analysis, we plan to combine with a lower resolution on the food habits and the anthropological sides. So we would reduce the data sets on both sides, but yet having a sufficient degree of preciseness. We submitted it to the FWO, and the project was evaluated as groundbreaking and necessary, but very high risk, and we were thus unable to secure the funding. So the contradicting demands of the disciplines to be wet were apparently hard to solve convincingly, not for the reviewer, but for the FWO. So since there is so little epigenetic research on non-Western populations, it is also impossible to use existing data sets for comparison. That would have, of course, eased our case. So it is true that the first, so it is true that the first attempts are essentially fishing expeditions where a great deal of luck is involved. So in cooperation with people from ESPOL in Guayaquil and ICIAM University in Tena in Ecuador, Wim Van Daele initiated a new interdisciplinary attempt to study the effects of breastfeeding practices on the epigenome of babies. And so I went there last August, not last, but the last August before the corona pandemic struck, to stay with the Amazonian Quechua Indians and to study the food habits there and how the food habits would affect the composition of the breast milk, which then shapes very much the epigenome as well as the gut microbiota of the infants, because at that time it's very malleable. And I also trained people at ESPOL in the art of food anthropology so that they could continue to pursue that work, because food questionnaires tend to be quite unreliable. But extensive ethical approvals have delayed the project, and we were starting normally with the data collection in March, but we have to wait until Corona is over, because that laboratory you see here had to be completely transformed for intensive Corona testing. So this is to be continued. So meanwhile, I also started reading about gut microbiota and the microbiome. The human gut microbiota consists of trillions of microorganisms, including bacteria, archaea, and viruses, who play an important role in food digesting, harvesting energy, and regulating immune development. And they're increasingly being acknowledged for their roles in regulating metabolism and several diseases. On top of the different roles it plays, it is also very heavily impacted by lifestyle, sleeping patterns, food, eating, medicinal systems, and such as antibiotics use, as well as pollutants in the natural environment. So here again, we see a clear responsiveness to the biosocial environment, which makes it interesting to force a cooperation between microbiologists and social scientists. It also calls for a food systems microbiome approach, because all the parts in the food system, from production, processing, preparation, and cooking, all influence the microbial composition of food, which affects and is affected by the gut microbiota. So I contacted the RAS lab at the Rega Institute of KU Leuven to start working together, where we would join forces in understanding how socio-cultural habits and natural environments ingested through food are entangled with the composition and the functional variation of gut microbiota among different human populations. And here again, we are talking about fishing expeditions, because most research on gut microbiota stems from research on white Western subjects in specific contexts. Although there are a few cases, a few rare examples of gut microbiota research, which is conducted with people in very different contexts. And I just discuss one here because it's a very interesting example, and it also is an example of how such interdisciplinary cooperation can mutually enrich the disciplines involved and lead to a more integrated approach to human health and life in terms of more than human health. So for our purpose, I just introduced the key bacterial lineages, which entail actinobacteria, bacteroidetes, firmicutes, proteobacteria, fusobacteria, and ferruciomi microbia. And it's especially the firmicutes and bacteroidetes that account for around 90% of the gut microbiota. But that figure then, the question is whether this comes from the Caucasian white subject. We don't know. So the study I look into here is a study published in 2014, and it conducted research on the gut microbiota among the Hadza hunter-gatherers in Tanzania. And they compared the data of the gut samples with urban Italians and rural farming groups in Malawi and Burkina Faso. And so, interestingly, for the first, they found that the overall diversity, biodiversity in the gut was bigger than the Italian sample. But it was clear that the Italian sample reflected the Mediterranean diet, which is heralded as being a very healthy diet, with a lot of plant food, fruit, pasta, bread, olive oil, low and moderate amounts of dairy products, chicken, red meat, and fish. Whereas the Hadza gut microbiota reflected the more plant-based diet for about 70% of the overall diet, but that includes then a lot of baobab, berries and tubers, and also some honey. On the meat side, which is about 30%, it's birds and game meat. So the microbiologist reads in this that the Hadza microbiome is much richer in proteobacteria and spirochaetes and have a higher presence of bacteroidetes compared to the Italian example, where then they found a higher presence of firmicutes, actinobacteria, and bifidobacterium, which is a suborder of the actinobacteria. And so the latter two, actinobacteria and bifidobacterium, were completely absent among the Hadza. And this is explained because they don't have, after breastfeeding, they don't have any contact with milk or cattle who gives milk. So you see the environment really shapes the gut. And so compared to the rural communities in Burkina Faso and Malawi, there is a higher presence of treponema and unclassified bacteroidetes and ruminococci among the foragers, because those bacteria enhance the ability to extract nutrition from those very strong fibrous foods like from the baobab. But then interestingly, they also read socio-anthropological accounts on the Hadza, where they read about the gender division of labor, where women forage for tubers and plant foods and stay at the camp with their children, and the men forage farther away from the campsite and also hunt for game meat and honey. And so that they hypothesized from that socio-anthropological description that might have a big impact on the gut microbiota composition. And so they divided the samples between men and women, and then they saw the gender statistical difference between the genders in terms of the microbiota composition. So the anthropological observations allowed them to refine the hypothesis, and by comparing the data in this slide, they were also not only able to show that the gender difference was there among the Hadza, but also explain that these differences reflect differential contexts with different environmental microbiomes. And this clear already indicates an advantage of such radically interdisciplinary cooperation. But then also on the larger timescale, they also found seasonal variations, because there are differential eating patterns according to the seasons. And that also spurred a follow-up study to see if the seasonal variations in food habits also was reflected in the gut microbiota. And yes, it did, because during the dry season, the Hadza live near the water holes. There is not much plant food together, so then they eat much more game meat, and the game also collects around the water holes, so there they can hunt and have more meat. So you see an annual variation in the gut microbiome, and this is a cyclical annual reconfiguration. So some taxa disappear during some time, only to reappear in a subsequent season. And this, in the literature, such concomitant seasonal variation in diet and gut microbiota is linked to an overall increased richness of the microbiome, which is good for overall health. And it shows how malleable and environment responsive the more than human becoming is. And this calls for an eco-health understanding. So through understanding of the both biological and socio-cultural processes and forms of regulation is required. A thorough understanding is required on both sides. And the last account on the Hadza, gendered and seasonal variations in eating and ways of living have shown that one understanding in one realm enables a better understanding in the other. Second, this cross-cultural and interdisciplinary understanding requires to put into question the universal Western metrics, the dominant health metrics, which is based on Western samples and which often determine how the WHO, World Health Organization, would work, and all the other standards by which we consider people healthy and also their microbiota healthy. So it's really necessary to decenter, decolonize, pluralize the human body and processualize and situate biologies. So from this small sample in this research, I thought, okay, this is really fascinating. So let's put up this interdisciplinary cooperation together with RASLab. And so we would there also try to aim for an equally high resolution on both the microbiological side as well as the socio-anthropological side, so that we could reach more precise correlations between social factors and food habits on the one hand, and composition and functions of gut microbiota on the other. Nowadays, all too broad categorizations are used in the statistical analysis, whereby the determining categories are very too broad and too internally diverse. Let's say like Western diets, industrial diets, ancestral diets. These are very broad categories with such an internal variation that the internal diversity within such broad category may outweigh the variability between the categories, and thereby inhibiting the detection of statistically relevant relations between environmental factors such as diet and the gut microbiota. So we need a higher resolution on the socio-anthropological aspects of food and eating than is common, precisely to allow tracing the biosocial pathways that affect specific gut microbiota. And so they have accepted the interdisciplinary adventure, and we are designing projects together to date. And an important aspect of this is the role that each discipline would play and the relation between them, affecting the ways in which forms of knowledge get subsumed, or when we want to achieve an integrated worldview, how we can work together. So there are many forms that such a cooperation can take. And the first broad category is where disciplines play an additive role, staying within each disciplinary boundary and where findings are added up, but not read diffractively. This is the kind of cooperation that is really the most common. And but still it involves many possibilities and exciting ones, such as the social study of the implicit conceptualizations of the human subject and the human evolution as held by social anthropologists, scientists, microbiologists, and the ethical implications of this conceptualization, or the social imaginaries and narratives that gut microbiota furnish, including the microbiomania, which is currently raging in different sectors. or new economies that emerge from the commoditization of gut microbiota. You can find an interesting website on biomerestoration.com. The intersection of microbiota and environment governance, because of the different pollutants and microbiomes in the environment. Relations of people with their microbiome and with each other. At a recent talk at Goldsmiths, Prof. Jamie Lorimer showed a blog called Colon Comrades. Microbiome, citizenship, and the individualization of health responsibility is another. So this is one category of cooperation, which is exciting, but I want to go a bit further and really be engaged in the very science of it, starting from the design of the project and where we really can learn from each other and be affected by each other through the diffractive readings of one through the other. So such an agenda would include, for instance, how culturally informed food habits and eating patterns, as well as the particular food systems and the microbiomes construed around these, are actually shaping the gut microbiota. Or electrification and food technologies, because fridges and freezers, they do have a fundamental effect on the kind of food we eat, ice cream and other things, and environmental pollution, purified water, antibiotics, human animals relationships as shaped by food systems, but also by religion and worldviews. So even worldviews through food co-constitute the gut, and therefore are constantly part of the more than human becoming. But again, this is not so easy. It might look fancy in theory, but for our project in Sri Lanka, where we will look into the role of environments and religions impinging on multiple kinds of engagement with food, and that will shape the gut microbiota in different ways, we have to find ways to integrate sometimes contradictory disciplinary requirements of obtaining detailed data sets from metagenomic shotgun sequencing of stool and multiplied by the enormous ethnographic information, all to be joined together in biocomputational analysis and contextualization. Another risk is that we molecularize, for facilitating such cooperation, that we molecularize the worldview and the biosocial environment, which then leads to a scientific reductionism of the broader multidimensional context. Even though such molecularization facilitates the scientific experimentation and examination, it is not apt to study the broader context shaping these proximate molecular agents affecting the gut and health. Let's think of the political process on regulating pesticides and the society's habitual use of these toxins that affect epigenetic and microbiome processes. So this means we have to find not only the molecular impacts, but also have the wider context included in this research, political, social, economic, and other contexts that are not reducible to molecules, but that do, however, take part in shaping interactions between the biosocial environment and the gut. So how to study these both aspects, the microbiological and the environment, as they converge in the more than human becoming, how to study these both sides which are in a constant flux. And we need to think of focusing on bodies in action, Jörg Niewerner argues, as a both basic unit of analysis and that understands the differentiation, the ongoing differentiation of bodies and environment and microbiota as a patterning of material semiotic practices. This differentiation, in other words, is always biosocial, co-evolving with gut microbiota, food activities, biosocial environments. But such cooperation easily comes into different problems as well, as encountered with the epigenetic part. And he says, yeah, experimental designs that can control the increased number of variables by adding more and more detailed social factors is again very difficult to design. You can't have very detailed ethnographic information on thousands of people. So how do we find how many social factors to correlate statistically with the thousands of different classes of people, but then multiplied with the millions of microbiota, and how to find statistically relevant correlations within a reduced sample? So this is the same problem as in the epigenome project. And also, as mentioned earlier, some of the variables are just difficult to quantify or typify. Cultural evolution, or policy making. And also the human lives themselves are enormously diverse, always in a state of becoming, and they also respond to changes in the environment, which they take part in shaping. So this makes the modeling of human environment systems a key challenge in getting a grip on its analysis. And there, I think complex system theory and cybernetics have an important role to play in the complex system biologies or complex systems microbiologies. So this would be an important part to develop an approach to food systems and food systems microbiomes. And also, there is social sciences and biological sciences have not really history of cooperating between the additive mode, where the social science views the body as something to be narrated, expressed through language and symbol. So the key challenge for the involved disciplines is to engage the other dynamic in the research of the biosocial entanglements and material semiotic processes that take place in environment-gut interactions. So Jörg Niewerner suggests to subscribe to a shared process and multiplicity ontology, where we focus on human becoming, as Tim Ingold would say, and how it is made and remade in a situated set of biosocial practices. I would call this an ontogenetic approach, where human becoming and health are situated and emergent within a material semiotic or biosocial network, entangled with both the environment and the gut microbiota, with the assemblage of food playing a central role here. So how to practically study such heterogeneous co-evolutions in the more than human becoming as a holobiome? First, we need to determine the level of detail on both sides, to say it like that, which would be sufficient for detecting a wide variety of species in the microbiota, as well as of giving a nuanced understanding of the lifeways and food practices of the human person. And where we agree on the similar level of resolution between the social and biological, and the degree of molecularization of the social. So these are some more practical questions. Second, given the demands for high resolution data in both levels, it seems that actually longitudinal research designs are best suited, given that you then can leave out a lot of intrapersonal variations, but you still can collect a lot of data within a small sample of people, thereby limiting these genetic factors that could create noise when having larger cross-section of samples. Also, a longitudinal approach makes it more compatible with in-depth interviews and careful anthropological research. Yet, a problem for some may be that the representational power at population level decreases. But still, it could allow us to understand better the dynamics. I just want to point at one exciting research where Lawrence et al. Collected stool samples from person A and B on a daily basis during an entire year. And so there you could see that when person A relocated to Southeast Asia for around 2 months, the samples showed very big changes in the microbiota community structure, connected to the altered food habits. And when he returned home, the balance was restabilized into the former state. Whereas in B, he got infected by salmonella, and that one has created persistent changes because it was the colonization of a new enteric species that reduced other species in his gut. So it shows that the Euro-American gut community can be altered, co-responding through shifts in food, environments, and context, indicating the situational malleability of human biology. And third, as food has such a powerful effect on gut microbiota, and as we ingest the world through our food, we take food and nutrition as the guiding lines in the networks and entanglements they consist of. We follow the socio-cultural and political economic aspects of the food system and eating, and the ways in which these shape the intake of macro and micronutrients on the one hand, and the ways these components relate to the gut microbiota on the other hand. And at a later stage, we hope to further pursue also the connections with health. Now there is a lot of research on one particular nutrient and effect on gut microbiota, which have done on the role of specific microbiota and health, but the more complex systems microbiology is still far away. And so, on a more fundamental note, to conclude, such cooperation requires a diffractive reading of microbiological and socio-anthropological work to arrive at the microbiosocial biographies of co-evolution. A focus on situating biologies, focusing on eating bodies as entangling with biosocial environments, bodies, nutrients, and gut microbiota. So to conclude, through the ever-responsive gut microbiota, the eating human being is a more than human becoming, co-evolving with ever-shifting biosocial environments through which health is enacted. This exploration of the complex biosocial network of food and food systems in relation to this in becoming and health is normally to start in Bhutan and Sri Lanka. And now I'm trying to speed up the project in Norway because other projects are on a standstill due to Corona. So thank you for your attention. And
thank you very much. Very interesting indeed. Very, very interesting. Very interesting indeed. I really was fascinated with this connection of anthropology to well-being. I think it's really very creative connection to health, and I think that provides I think a lot of space for thinking. I'm still digesting what you presented, so I'm processing. So if anyone has some feedback or question or anything.
Well, I'm speaking in the most general terms about interdisciplinary studies of complex systems, which is this definitely is. And I would say that there are, in a sense, two dimensions on. The one is to really look at the complexity. That means that you have lots of different factors that have lots of different relationships. And then you will need to make some kind of a model that will be a computer model, because it's becomes simply too difficult to do that with a traditional mathematical model or conceptual model. And there, one of the possibilities that I wouldn't immediately know how to apply it is the formalism that we are working on in a different project, the Templeton project, chemical organization theory, which is a formalism that tells you how a combination of factors leads to a combination of factors, which leads to another combination of factors, and the whole thing can become autopoietic, that means self-maintaining.
Yeah.
In this case, you're speaking about different bacteria, which consume, I suppose, different parts of the diet and which produce certain other molecules in the body, which may have certain effects on the body, which in turn may affect maybe either the food or the microbiota. So I think theoretically you see some kind of a network of self-maintaining factors. So possibly if there would be enough data collected, I suppose that there will be some methodologies to turn this into a reaction network. Now, I am not really the specialist on the modeling of that. For that, you would have to ask our colleague Thomas Velos, who is working with those things. But I know that they have been using these kind of models for metabolic modeling, also for modeling of social and ecological interaction. So in principle, the combination between social, ecological, and molecular things should be able to model in that. So that is one aspect, which is looking at the details and trying to make a complex model of it. But the other aspect, which in a sense interests me more, is to make abstraction of all the details which are different from every person and from every moment, and look at the global factor of what we may call health or well-being. And there we are also using another concept in our project on Templeton, that is that you have these very specific networks, but these networks can be characterized by something that we call resilience. So the resilience is the ability of the network to adapt to all kinds of changes and perturbations. So I would define health in this case as the adaptability or the evolvability of the whole network, so that if, for example, you put in a perturbation like a disease like salmonella, let's say, or you take away something, for example, suppose the person normally eats a lot of rice and for whatever reason there's no more rice available in the country, so he can't eat rice or he can't eat meat or whatever, then the healthy system would be the one that adapts. So I wonder, I mean, if I'm thinking in terms of microbiomes, I'm not a specialist, but I would think that one factor probably is the diversity of the microbiome. So at first sight, you would expect that the more diverse the microbiome is, the more resilient it will be. That means the easier it will adapt to changes in circumstances. So maybe you can comment on that.
Yeah, you have friends and foes in the gut microbiome. So you also some may be good. It's different aspects, because what you see, they call it the gut microbiota in deficiency syndrome, where the overall diversity is decreasing, and owing to an industrialized diet. But then again, that is too imprecise. What is an industrialized diet? So there, I think it is important to really specify sometimes, so that we don't confound different parts what is the cause there. On the other hand, there is also the gut microbiota. There are different layers in the gut, and so one can be a friend when it's in one layer, but when it moves, when there is a leakage to another layer, it can become a foe. So it is also very place dependent on how a gut microbiota can be playing a role in the overall picture. And then it's, yeah, and of course, some of the diversity can be also detrimental. So it's not always that all diversity is good. So these specifications are quite important in terms of place, location. Also, even microbiota, they may be the same, but may have a different functionality in some cases where they perform a different function. So there is again the potentiality. One microbiota can do some different things according to a certain situation. So I think this is a extremely complex system with trillions. And that's one of our key challenges, how to then relate this to the enormous complexity of a human life. Because we know all these different aspects, they do affect what happens inside, and vice versa. We are, I mean, they call it sometimes the second brain. So when we talk about gut cognition, in a way, what I mean, yeah, I think there is still a lot to be discerned there. Also, the gut-brain axis, what they call it, in terms of schizophrenia and depression. There are they know correlations, but causalities in which direction, it's not yet known. So it's, I think there is a lot of interesting things to be pursued further there. But I think your comment on the complex modeling, I think it would be very useful to think further about that, because that would maybe also enable how we can think about the biocomputing as well. Because that is going to be entirely new, because we haven't found research that has been doing this in practice. So this is kind of, yeah, we are trying out something very experimental and high risk, but yeah, we'll see. Okay. Thank you very much for the comments.
Very interesting, Wim. I talked a lot with Wim, that's years ago, and then he was more still, but I see that you moved really to more the medical aspects of food also.
Yeah.
And I read occasionally on that, but of course not enough to know so much about it. But I indeed, as far as I remember, I don't know if that's really serious, but even diseases like Parkinson and so, they are connected. They seem to be connected to the health of the gut. That is what I hear and there read. But it's very complex, of course, and that's also true.
Yes, I mean, following that, I wanted to ask Marius, if you're, I don't know if you are hearing me. I wanted to know your opinion on this, the composition of microbiota, and also the connection between cognitive degenerative diseases. And also, I think Wim just mentioned the brain and gut axis and the blood-brain barrier. If you have any thoughts on this connection, that if there is anything that you can share about what we mentioned about the difference between the diet in Africa and Italy. Marius?
Sorry, I tried to unmute myself. Can you repeat the question about the blood-brain barrier?
Yes, I mean, I was really curious about this biodiversity research of that Wim just mentioned about the connection between brain and barrier. Just a moment, I have to, just a moment.
Yeah. Well, anyway, the blood-brain barrier is a fascinating thing for me because I see it as an interface between the brain and the environment.
Okay, sorry. The connection between blood and brain barrier about this biodiversity of Italy and Africa, the research that Wim just mentioning, I was thinking that is there any, like, can we make some connection between blood-brain barrier and also cognitive degenerative diseases? Right now, there is a lot of literature and thinking about how the food we eat really impacts brain and how we can change our lifestyle and influence the resilience of the brain.
Well, the thing is that the food that we eat is not only acting through the blood-brain barrier, but it's acting through the gut microbiome. So it is a proven fact that certain foods interacting with the gut microbiome can create substances like serotonin, which can affect the brain. And so, for example, depression can be influenced by the gut microbiome. Also, there is some research about Alzheimer's that can be connected to this. It's not very clear how, but in my practice, I pay a lot of attention to the microbiome. It is something that surely it can affect the brain. It works. It's not only affects the brain, but it works in tandem with the brain. So it's like we are talking about second brain here. One influences the other. So I think this is just a very fascinating area to study. I'm trying myself to go deeper into this, and I discover new things all the time, interesting things.
Yes, I think it's indeed very interesting, especially I'm quite fascinated by the anthropological approach to well-being, and also how this would and could impact the way we eat in future, especially with this biocomputing that was just shared. So I think the future of well-being definitely would have some connection with technology and even how we, for example, when you speak about microbiota, you're speaking about the food that is produced naturally in nature, right? So what?
No, it's not that. Microbiota are the bacteria that live in our gut, in our digestive system.
That I know. The impact that it has, the changes that they experience is based on the food that are cultivated in nature, so the food we eat. But what will happen, what are the changes with the food that are designed in the lab? So
I don't know. I think nature finds a way to deal with natural and synthetic foods. it's my opinion that even if we have something synthetic and the body will try to use it and utilize it in a manner that won't harm us. And if that goes out of control, then it will produce some kind of illness. But I trust the way nature works.
That's one of the things that was shown, that which is a discussion with the current gut microbiome deficiency syndrome, which is very common among Western populations, is that our epigenetic system is not yet adapted to the sudden change in processed foods. And that's why our gut microbiota is not yet able to recover from that industrialized food and processed food. So because evolution takes place on many time scales as well, and apparently one of the time scales is lagging behind another, which is kind of, yeah, maybe the gut microbiota are adapting a bit faster, but our genetic system is not yet adapted. So the gut microbiota deficiency syndrome can lead to, therefore, to diabetes and obesity.
Well,
so,
I can mention something here, an example to think about is that as we use more our brain, now our society is becoming more cognitive. So we use the brain more than we use our muscles. Therefore, certain foods that people think they are bad for us, they may be actually good for us, like sweets. If we are using the computer a lot, many hours a day, then we need ready-made fuel to make our brain work there and then without depending on other metabolic processes. This is one idea. It's not that I support it.
Yeah.
One idea to discuss.
But it's indeed true. Some of the things we ate, of course, the connection with so-called lifestyle, but I don't like the term that much. But in medical sciences or nutritional sciences, it's talk about lifestyle, but yeah, ways of living which are changing, and therefore also our metabolic household is changing a bit. So and there I can see that some foods are better suited, but my interest actually now lies more in the fact that a lot of biosciences are focused on the white Caucasian subject, and I read somewhere that 98% of the researchers in biomedical research on persons, on human persons, is done on white Caucasian subjects. So there is a problem with using the white body as the metric for the whole world, where we can see that bodies are partially enacted by the environment. And also, of course, some genetic differences as well. So that's why I want to, my aim is also to kind of situate biologies more and decentralize it, to decolonize it in a way, to say it as an anthropologist, and precisely by looking, for instance, the project which is now under review for application here at the Norwegian Research Council is on Bhutan. And there, for instance, we will carefully focus on 10 people, where we will follow 10 people very closely. So every day we will we will measure what they eat, but also have in a lot of talks about what they do. We will have observation in the village where I will stay. And then we'll try to first calculate what the, we'll calculate what, for instance, the effect is of a ritual on micro and macronutrient intake. And we'll also take a stool sample every day. So it's going to be very intense. I will do it myself too, because I'm also traveling around and taking also that as a clue. But this is kind of a starting up, and then hopefully we can later apply for larger funding for more cross-sectional, larger groups of people. But I want to understand the local biology of a person, a yak herder in the Himalayas, who is eating a lot of yak products, milk, cheese, butter, and meat, versus a Sri Lankan Muslim fisherman who does the Ramadan, and how does the Ramadan affect it? So to understand really that interplay between cultural, socio-cultural, religious habits, as well as our biology, how they co-evolve.
The question that is related to what you were saying, you have been looking into hunter-gatherers. There is this big divide between people who are interested in diet, between more paleo type diet, where the idea is that genetically we are still pre-agriculture. And then there is, of course, also the diets focused on natural agriculture, which are very different. One is really a very different diet than the other one. With what you have been seeing now in more detail, do you see some systematics in there, or is that, I mean, do you see some, who is more right, the paleo people or the vegetarian people, the agriculture people, let me say it like that?
I just have a question, because we know that with the domestication of fire, we got them domesticated by fire, in the sense that when we started cooking food, it had enormous consequences on our brain development. It had consequences on our gut development. So we have been changing because we started cooking foods, and we started eating more diverse, and grains are also very useful for energy, and that energy provision was no problem until recent. But then also we have, of course, included or added a lot of added sugar, added fat, and so on. But yeah, so I think we have, so my hunch is that we have developed so that a paleo diet might have some interesting and healthy aspects to it, but not entirely, or not entirely suited to our bodies today. Maybe for some peoples in the world. I think the plan is also to do more research in Amazonias. So we'll see what happens from there. But that's also kind of looking at some groups that live quite detached from industrialized foods. So to see how their gut microbiota is, and epigenetics is. But I'm not a specialist on the paleo diet, so I can't say much more
No, but let's say it's more the idea that very different. If you make reference to the pre-agriculture period genetically, let's say, or if you think that our genes have co-developed and actually already or in more agriculture period, then this in the industrial period, that I think is quite clear that this is very short still. So we probably have not genetically adapted to that.
I think, there is, the genetic adaptation is on a different time scale than the epigenetic adaptation and then the microbiome adaptation. So that's where you have a mismatch in different forms of adaptation, which is interesting in itself to think about resilience and multiple forms of adaptation.
Yeah. But again,
one adaptation can make the overall complex system maladapted because they're not yet in tune with each other and not in tune with the changing environment. So,
I don't know if Francis, are there clues out of pure evolutionary biology on this question, whether genetically we are still closer to the paleo type or to the agricultural type?
Well, I think that logically you would expect we have been evolving for millions of years on a paleo diet. We have been evolving for maximum 10,000 years on an agricultural diet. You would expect that the agricultural diet has had some influence, but not as much as the paleo diet. So I would expect that we are probably better suited to the agricultural diet than our ancestors of 10,000 years ago. But we're still closer, I would say, to the paleo diet genetically than we are to the agricultural diet. Also because these 10,000 years, that's 10,000 years, the beginning of agriculture in Mesopotamia, in most parts of the world, agriculture started later. So in a couple of thousands of years, there will be changes. For example, one of the famous changes that is well established is that many populations can digest milk, which others cannot. Milk, because part of the agricultural diet is milk-based, but for example, in China, most people are not capable to digest lactose, and so they would feel pretty bad if you give them cheese. So that is one example where you see there is an adaptation, but you see the adaptation is not universal yet. So I would assume that for other things it will be similar, that there will be some degree of adaptation to grains, but not that we are perfectly adapted to grains. And actually, I personally, I don't think that the difference is that much between a pure paleo diet and the agricultural diet. I think the difference is between a more paleo style diet and an industrial diet. And the industrial diet is one with highly digestible, very nutrient poor things like sugar, pasta, fries, hamburgers. That is a problem. In the agricultural diet is already more like that, but yeah, if you go back to a diet that is more nutrient dense and more difficult to digest, I think it will be healthier.
But also the animals have evolved from that time. The animals that were eaten in paleo diet. So there is also an evolution. So we don't have exactly the same animals than them. But
I don't think that the protein, etc., composition of the animals is that big. The animals now may probably have more fatty meat. That will be one difference. But otherwise,
yeah, that's also why we want to include the gut microbiota of animals, because we share a lot of them in interaction. So my pilot in Norway is really about reindeer herders who live together a lot with reindeer and who eat a lot of reindeer products. And then to see also the seasonal variation in that, because they move from one place to the summer pastures and then back. And yeah, so because we see that people and animals co-evolve as well, especially those who are in close contact. So there is, but then the domestication of fire has also made a lot of changes physically. So it's maybe not genetically, but then it's something else.
Well, fire is probably at least 1 million years old that we have discovered it. So you would expect that fire has had a much bigger influence on agriculture. And
Yeah.
fire means things that are cooked, they are much easier to digest.
Yeah,
like,
Ryan
to be able to digest leaves the way that gorillas eat leaves, definitely we wouldn't be able to do that anymore.
Yeah, yeah. Reinham has written, I was trying to find the book, but I can't see it immediately. Yeah.
I wanted to ask a different question. We have now mostly been speaking about the microbiome, but you started with the epigenome. And there's one concept that I have recently encountered, but I haven't yet read a lot about it, and that is a so-called epigenetic clock. So it's a theory of aging that as you age, your epigenome starts changing in a way where it becomes less effective, and that by measuring the epigenome, you would have an idea of how much somebody has deteriorated because of aging. And so I was wondering if you can measure this epigenome in this way, and if the epigenetic clock would be some kind of a measure of deterioration, that maybe it could also be used as a measure of health of the epigenome, and that you could, for example, compare the epigenome of people on different diets and using something like the epigenetic clock to see which one of those is healthier in the sense of closer to their youthful appearance and less deterioration.
Yeah, Wim Van Daele would be really good to answer that one. He's also looking at aging and inflammation actually. So it's really about how a lot of inflammatory diseases become more common when you start aging. So that's where you draw it together, inflammaging. But then it's also I think I read about the epigenetic clock in different ways. But Wim van den Berg could really be very useful on your question. But there is another thing which is about the metabolic clock, which is also epigenetically regulated. That will say, for instance, how you eat regularly according to a similar rhythm. And that rhythm is important because when you constantly shift the times when you eat, then the metabolic clock, which is epigenetically regulated, can get disturbed, and that creates metabolic leakages through which the regulation of fat and energy and sugar gets disrupted, and that is also part of the whole obesity and diabetes problem. And of course, with the 24-hour economy, that has an impact on these metabolic leakages. I'm now a bit out of the epigenetic literature because I've been focusing on the microbiota literature more now. But I'm also planning again to work together with Wim van den Berg again on trying again with a different research design than last time. So we'll see if we can make it there.
Thank you very much, everyone, for today. We're at 6:30. Is there any final question, something that needs to be shared, or something final, or some final notes? Because I have to close the meeting for today.
I wanted to thank you all, and it was a very interesting discussion afterwards. So I would be interested in contributing if there would be an application or something then. Yeah.
Thank you very much, everyone, for being with us today. Have a great evening, everyone, and goodbye.
Yeah. Bye.
Bye.
You.